Hello, and welcome to our Structural Biology Behind Disease Transmission and Drug Design panel discussion. My name is Dr. Sharon Pepenella, and I'm an educator at the Cold Spring Harbor Laboratory DNA Learning Center. With me today is my co-host and panel moderator, Dr. Aleida Perez, an educator in the Office of Educational Programs at Brookhaven National Laboratory. We are excited to present to you this event as a follow-up to the premiere of Branching Out with Bio, the third episode in the Full STEM Ahead Long Island educational video series, which introduces students to STEM education and career opportunities across Long Island. This series is a collaborative effort between industry, academic, and nonprofit facilities who are part of the Long Island STEM Hub organization. Before we get started, we'd like to remind our audience that you can submit questions to us during this panel discussion using the BlueJeans Q&A section. Please know that while you will not see your question appear in the Q&A dialog box, it has been received by our moderator. We will do our best to address as many questions as possible. If you are having technical difficulties with the stream, you can let IT know by using the chat feature. The Branching Out with Bio video, which we hope many of you had the chance to view ahead of this panel discussion, highlights structural biology research conducted by students and scientists at Cold Spring Harbor Laboratory and Brookhaven National Laboratory. Our panelists today, who are researchers from these two facilities, join us to discuss how they use structural biology in their research, the importance of understanding the structural biology behind disease transmission and drug design and how that applies to our current pandemic, and career paths for STEM enthusiasts. Panelist Dr. Alex Suarez is an X-ray crystallographer for Brookhaven National Laboratory who uses sound pulses to levitate small amounts of drug candidates and protein targets, miniaturizing the scale of experiments in order to maximize the number of compounds that can be tested. Panelist Dr. Jonathan Ipsaro is a structural biologist from Cold Spring Harbor Laboratory who studies the molecular machines that guard our genomes against deterioration by obtaining high-resolution, three-dimensional images of those guardian molecules. Welcome, Alex and John. So Alex, why don't you tell us a little bit more about yourself and the structural biology work that you do? We can hear you.
Okay, so I started structural biology as a graduate student, mostly because I was fascinated with the interplay between structural biology and drug development and health issues. But very soon it became apparent to me, even as a very young graduate student, that most often the limiting factor was the amount of available sample. This was back in the early 21st century when we didn't have the same techniques as we do today, both in terms of beam size and in terms of sample prep. So I focused most of my career on reducing the size of biological experiments, reducing the aliquots necessary of both protein and of tested chemicals and tested materials, so that we can do a wider range of assays and structural biology experiments with the same starting amounts of materials. So this is the acoustic sample preparation technique that I've used throughout my life, and I'll talk a little bit more about it later.
That's really interesting. Thank you. John, could you provide us with some more background about yourself and your research?
Yeah, so thanks so much for having this, by the way. I think it's really great to be able to at least virtually interact with people right now. So, when I was going to college, when I was a high school student and trying to figure out what I wanted to do, I always knew I wanted to do some sort of science. But I actually went in thinking I was going to do computer science. But then when I started taking some, kind of obligatory biology classes, I realized there's some interesting stuff here. And then when I was introduced to structural biology, I realized oh, wow, you can actually see things in 3 dimensions, zoomed in at these crazy, amazing levels. It's like you're in outer space and you're looking at leaves on a tree on Earth, right? It's this crazy amount of zoom that you can get to really understand how molecular machines are working. And so when I saw that, I knew right away that was the type of work that I wanted to pursue. And that's what I've been doing ever since. So.
Fantastic, very exciting. All right, so I'd like to start our panel discussion today with kind of a general question for either of our panelists. So what exactly is structural biology and what makes it different from other subdisciplines of biology?
Sure, I'll start with this and then maybe Alex can chime in himself. So structural biology is really trying to understand how biology works at a molecular level by taking very high resolution, three-dimensional pictures. And I think one thing that's important to realize when you see some of the images that Alex and I may share with you today, that those images are not just three-dimensional pictures in the sense that you're seeing the outside of something, but you're actually able to see through something. So it's like having a photograph of a person and also an MRI and also an X-ray all at the same time. And this is being done at the angstrom level, which is 1 times 10 to the minus 10 meters. So, millions of times smaller than things we're used to seeing. And the idea is that if you can generate these high resolution three-dimensional images of molecules, you can get a good sense of how they work. So an analogy I always like to give is that, if you know how to drive, every year there's a whole bunch of new cars that come onto the market, and you've never seen that car before. But if I gave you the keys, you would know exactly what to do. You would know how to open the door, you would know how to get in, you would know how to put the keys in, and you would know how to go. And because we as biologists have been studying proteins and nucleic acids for so long, we have lots of pictures, we know lots about how they work, and so with new pictures, we can deduce a lot about how they might be functioning. So that's structural biology to me.
Fantastic. That's a really good analogy. Thank you. Alex, do you have anything you wanted to add to that?
Just a quick note that was imprinted on my mind by my original graduate school advisor, Donald Casper, who coined the term structural biology. When he first used that word, he got a lot of pushback from people that said, you should call it structural molecular biology. But he insisted this was not a good idea because as time went by, we weren't going to focus only on static views of molecules, but we were going to focus more and more, as techniques improved, on a dynamic view, which has a time axis. And so he didn't want the word molecular in there because he didn't want people thinking about it as a picture. Well, for a lot of time, that didn't happen. But recently, and Don Casper is still with us, by the way, he's 96 years old right now, but recently, techniques have advanced sufficiently that we're now able to produce molecular movies. We no longer give you static pictures of what a molecule was doing at a particular point in time. We give you dynamic movies of what it's doing throughout its life cycle. So Don Casper's original vision for structural biology is really getting realized now as we begin to have techniques like cryo-electron microscopy and advanced X-ray crystallography that allow us to make those molecular movies.
Wow, that's fascinating. So does that mean we're able to then see the movement of proteins and kind of looking at what happens when something binds to an active site and causes the protein to maybe change shape? We see those kinds of things now. Fantastic.
Using John's car analogy, it's the difference between looking at a car in a magazine and seeing a car driving in front of you. You get different kinds of information from both those two things.
Great, thank you, Alex and John. So let's, John, we'll go back to you. So the video shows viewers a few different kinds of techniques that scientists use for structural biology research, one of which is cryo-electron microscopy. So could you speak to what electron microscopy is and what the difference between electron microscopy and cryo-electron microscopy are?
Sure. So I'm going to share my screen really quick. And sharing. Perfect. Yeah, so, I think a lot of people have probably seen some images like this tardigrade that's in the upper left-hand corner of your screen, right? And that's an electron micrograph, but so are all of the images. Actually, I don't know if you can see my pointer, but this guy is, and then there are these two fields of particles here. Those are both electron micrographs. So first off, as you can guess from the name, the difference between electron microscopy and normal microscopy, which can be called light microscopy, is that you're actually using electrons, you're using particles to do the imaging. Now, there are some big differences between these types of images on the screen. So if you look at our little tardigrade here, the way that image is actually generated is that you take your sample, you take your little tardigrade, and you coat him in a very thin layer, typically of some metal stain, and then you bombard him in a giant column that you can irradiate essentially with electrons. And what happens is those electrons are going to kind of scatter off of him. And from that scattering, you're able to basically figure out what the shape of him is. Now, what we use, though, in trying to determine the structures of molecules is actually a transmission mode of electron microscopy. And that's what these types of images look like here. So I should mention that the zoom on these images is several thousand-fold higher than this guy. But the difference here is that now, instead of kind of looking at surfaces of things, we are actually using such high electrons that they actually go through the sample and you measure on the other side. So just like you can think of if you're looking at a normal light microscope and you have some slide that you put on the microscope and you shine light from the bottom through the sample through to your eyepiece so you can look at it. This works in a very similar way. You're shining electrons from one side through the sample and then you detect on the other side of it. Now what makes something like this on the bottom left corner, this is called negative stain electron microscopy. This is not cryo-EM. The one on the upper right over here, this one is cryo-EM. And you'll notice that the contrast in those images is really different. And the reason why that contrast is so different is that these down here, like our tardigrade friend, actually have a thin layer of stain applied to them. Okay, so as biologists, we're typically working with samples that are in solution. But in this case, what we've done is we've actually dried those samples down and put a thin layer of stain on top of them. And that's actually what's giving us contrast between our proteins and anything around them, because the stain is very dense, so it's very dark. Oops. And the protein molecules are excluding the stain, so they come out very light. Now, cryo-EM is different. Cryo-EM, you actually just take the sample and you freeze it. You freeze it in place. Basically what happens is you take your little sample in a tube, you put it on a grid. These grids are about the size of a hole punch, and then you freeze it super fast. We actually use liquid ethane, which allows you to cool the sample even faster than if you use liquid nitrogen. So everyone knows liquid nitrogen is really cold. This can cool things even faster. And what that does is it freezes all of the molecules exactly in place. From there, though, the electron microscopy is largely the same. You're still going to bombard with electrons going through the sample, and you get these kind of particle images that you get where each one of these little dots is one of your molecules. Now, what makes cryo-EM so special is that you can get, first off, a lot of artifacts from staining. So our proteins don't live in stains of high amounts of metal in them. They live in more like a water-like solution. So that helps us mimic biology better. In addition to that, because of the less contrast that we get actually in cryo-EM, we actually get higher resolution information. So there's this trade-off in electron microscopy where the more contrast you have, the easier it is to see your particles. Like here, you can see that it's very difficult in the upper right corner to pick things out, but in the bottom left, it's very easy. There's this trade-off between contrast and resolution. And what we ultimately want is to be able to pick our particles and also generate high-resolution maps. And you'll see some images like that Alex will show you today. Hope that gave a good overview of the technique.
Yeah, that's great. And by rapidly freezing these samples, I imagine that it means that you're also getting them in a more natural state. So you're not changing the shape of the molecule, the protein, or the nucleic acid.
Yes, that's what you hope. So, you never know. And again, our proteins and DNA and things, those live inside of cells. And when we do these experiments, they're in a tube. So there can be some things that you have to watch out for, but we of course do other types of biochemical methods to make sure that what we're seeing at the end of the day actually makes biological sense.
Great. And cryo-electron microscopy may not be super familiar to all of our viewers, but it's certainly a star in the structural biology world.
Yes.
In fact, the scientists who developed cryo-electron microscopy received the Nobel Prize in chemistry in 2017 for that scientific advancement. And we're certainly excited to have a cryo-EM facility at Cold Spring Harbor Laboratory. So that's very cool.
Lucky. We're very lucky. It's a really cool facility. And I know the lab does tours when it's not coronavirus time. And they do show off the microscope. So if you're interested in seeing that, I'd highly recommend it.
Can you just give a quick example of the type of research that might be done with cryo-electron microscopy?
Yeah, so it's a really versatile technique. I think Alex might speak on X-ray crystallography more, which is also super versatile. At the lab, we've used cryo-EM to determine the structures of virus particles, for instance, including rhinovirus, which is like a common cold virus. It can also be used for determining the structures of single proteins, looking at how those proteins structures may change. In examples like you mentioned, like if you have a compound binding to them, or if you have multiple molecular components that bind to each other, to see how those affect each other. So it's really a tool for us. So, both the things that Alex and I do are really geared at how do we get at the images and movies of how all of these molecules are working, and we will use whatever tool we need to get those answers.
Okay. So another technique that was featured both in the video and John that you just mentioned is, of course, X-ray crystallography. So Alex, can you explain how X-ray crystallography works and what kind of information we can obtain from using that technique?
Okay, so a crystal is basically an ordered array of objects, identical object that's similar to maybe in your grandmother's bathroom, you might see tiles with pictures in them arranged right next to each other in an ordered array. That's what a crystal is. Why is it useful? If you direct a beam of X-rays through a crystal, what emerges is the electron cloud of the object that the crystal is made of. Not of the crystal itself, but whatever the crystal is made of. Not of the tile wall, but of the image within the tile wall, if you can use the analogy. Most of the times, 98% of all medically relevant biochemistry is carried out by proteins. So most of the time, when we're growing crystals of proteins. And the reason for that is because proteins are the things that make us well, and so when proteins aren't working well, that's when we become sick. And so what we're usually trying to do is find out how to make a defective protein work better, or in the case of an invading organism, how to interfere with the critical protein of that invading organism. So key thing to remember is when you put X-rays through a crystal of a thing, what comes out is the electron cloud of that thing. The physical mechanism by which this happens is a little complicated to explain here, and maybe not really that relevant. You can learn it easily once you decide to become a crystallographer. The key thing is, why is it useful? The reason it's useful is because when you put X-rays into a thing, what emerges is the electron cloud of the crystal that thing is made of.
Now, one of the questions that we got from a viewer was, how do we know that the electron reconstruction is accurate from this cloud that is formed?
I would say that question is pertinent to all structural biology methods. One of the key ways is because we have multiple panelists. So whenever we solve a structure using crystallography, if they solve the same structure using either cryo-electron microscopy or nuclear magnetic resonance, NMR, then we can compare the two structures, and if they're not the same, we know we have a problem. That really has never happened where they're very different. Maybe a couple of exceptions, but for the most part, they've been correct, which then shows that they're both right. The other thing we can do is we can compare the results that we get with other known information. For example, we know basic properties of chemistry and of physics that tell us what molecules should behave like and how they should interact with each other. And when we see that the electron cloud that we observe matches the electron cloud that we know we should observe, given the chemistry and biology and physics that we know, then we know we're right. For example, we know the sequence of the proteins that we look at. We know that from sequencing information. And then if we see that the sequence that we observe in the electron cloud is the same as the sequence that we expect, that lends a lot of credibility to the experimental method.
Great. Well, Brookhaven National Laboratory has a light source that scientists from across the country and even around the world come to use, which is the National Synchrotron Light Source 2, the NSLS2. So we've been told that this light source is so large you can fit Yankee Stadium inside of it. So why does it need to be so big?
The technical answer to that is that the bigger a synchrotron is, the higher the quality of the X-ray beam that emerges. However, I wouldn't call that a completely fair or accurate answer, because NSLS2 is a little bit bigger than it actually needs to be. For example, it's bigger than MAX5, a equally good quality X-ray developed a machine from Sweden. Beyond the technical answer that size gives quality, there's also an additional Walmart parking lot answer, which is that the bigger the synchrotron is, the more scientists can do simultaneous experiments because we can fit more scientific end stations around it. So in the case of NSLS2, the size is not really driven by the technical need for higher quality X-rays. That's part of it. But it's also driven in large part because there are just too many scientists shopping for solutions to too many problems to fit around the smaller rings, such as MAX 5 in Sweden.
Excellent. So, John, we'll come back to you. So you work in the laboratory of Dr. Limor Joshua Tor, who was featured in this video. And she discusses how her lab uses structural biology to identify a protein that actually helps destroy an RNA that, when it's unregulated, can lead to neuroblastoma, so a cancer that primarily affects young children. Could you elaborate on how something like X-ray crystallography was able to help establish that this protein affects that RNA and how this is useful for developing things like therapies?
Right. So I think one point I'd like to back up on here that really kind of gives a bit of perspective on how structural biology works a lot of time is that usually my job or Alex's job begins when we either read a paper or we have a friend or colleague who has some question that they can't answer with their normal methodology. So for instance, someone might be interested in pancreatic cancer or neuroblastoma or Alzheimer's or what have you, and they say, okay, we have found through genetic evidence or looking at patient samples or all sorts of different other subfields of biology, have found this really interesting gene that we know is super important, and we have no idea how it works. And that's really where structural biology gives you a lot of power. So in the case of the protein that Limor was talking about, the name of that protein is argonaute. And argonaute is really important for being able to do this sort of RNA, which is if you watch the video, is a kind of intermediate molecule between DNA and protein, and being able to regulate the amounts of that RNA that are present, and kind of doing that on the fly. So argonaute is a protein, and what was kind of known through some great collaborations actually that were happening simultaneously at Cold Spring Harbor was that, this protein really seems like it's really critical for allowing this modulation of RNA on the fly. But we don't know how it works, and we don't know exactly which proteins are involved. And so that's when Limor's lab stepped in, and they said, all right, let's try to determine the structure of it. Now, at that time, cryo-electron microscopy actually was not the star player that it has become in the last 5 to 10 years. So we used crystallography in that case. And what they were able to see when you get the picture of the car, in this case, argonaute, what you're able to see is, it really has a lot of features that would make it look like an RNA binding molecule. So RNA is negatively charged and the protein has a very positively charged patch. You look at particular areas of the protein and you realize it has similar, we call them domains, similar regions of the protein that have particular activities to other proteins that we know about. And when we kind of assemble all of that together, what you can understand is that this protein really looks like the one that actually does the RNA degradation itself. It has all of the components that are needed to actually do that part of the biology. So by starting off with some cool genetics and biochemistry from collaborators and knowing that this protein is really important in a pathway, and then taking a picture of it or a movie of it, or getting these types of graphics of it, you're able to deduce what its actual function is. And then we hand that back over to our collaborators and say, based on the structure, we expect that if you make these changes to it, it will no longer work or it will work better. And so you can then validate all of that in the actual biological system. So that's how it kind of like all wraps together.
Very cool. So we have a viewer question. I'll open up to either of you. So what role does supercomputing play in your work and how important is it?
This one's for you, John.
Okay, yeah. So it's really actually that's a really fun question to answer as a crystallographer and cryo-electron microscopist. So I started my training as a crystallographer, and that's actually still where I'm the most comfortable. And I think it's important to give credit to early crystallographers because, nowadays people easily work with like gigabytes and terabytes of data, right? But when crystallography was becoming a thing, actually, there were a lot of crystallographers that had to be involved on the computer science end of things to process data. Interestingly, though, the size of the data sets that we're working on now are relatively easy to handle by all of the amazing computing technology that we have, right? I mean, and a lot of that is not just due to technical advances in computing for science, but also, people play video games. And when people want to play video games quickly, you get great computers out of it. So supercomputing, in terms of where it actually is really critical in structural biology right now, is in electron microscopy. And the reason for that is that when we collect these cryo-EM data sets, if you remember those micrographs that I showed you where you had all the little particles on them, those are actually not still frames. Those are actually movies. And the way that they're generated is you have your sample in solution and you're beaming the electrons through it. That's a very thin layer of liquid. Okay? It's only like a micron thick or so. So it's very, very thin. And when you do that, that kind of shakes everything up, right? It starts melting the sample, things start moving. And you can imagine that if you want to be able to see something in very fine detail, you don't want it to be moving around, right? You always stand still for a picture. You don't start shaking around, right? You'll get a blurry picture. And so what those micrographs actually are we do a process called motion correction on them. So you actually irradiate, and while you image, you actually take a whole bunch of very quick, not stop motion, but the frame rate is very, very high, and you process those frames independently first, and then you compare each frame to each other to kind of rejigger where all of the particles are so that you can average them more precisely. That takes a lot of computational power. The frames themselves actually take up quite a lot of space. So right now at the lab, when we collect an overnight data set for a protein, so the microscope is largely automated. You get everything set up and then you click go and you hope everything goes well. If you collect data overnight for 12 to 16 hours, you'll end up with about 1 terabyte or 1.5 terabytes of data. And then that needs to then be processed. So you get up with these intermediate processing files. Each one of those movies that are aligned can contain, I'd say on average, between 50 and 500 particles. And you have to extract all of those and then figure out how they all relate in orientation to each other. So supercomputing, let's say powerful computing is very important. So we have a lot of GPUs in the lab. If you're a video gamer, you know that GPUs are good and good for parallel processing of things. Right now, we don't use a lot of supercomputing clusters. So there are some national clusters for supercomputing, or sometimes people will actually use Amazon Web Services for supercomputing power. And that can be done, especially if you're in a rush. But most of the time, we're able to process our data in a few days with just having a few really, really nice computers in the lab. But managing the data is really challenging. We have a lot of hard drives flying around.
I can imagine.
Yeah.
So let's go ahead and we'll delve a little. Oh, go ahead, Alex.
I think Alex wants to say something.
Very quick mentioning of crystallography. We don't have nearly the supercomputer needs that they do in cryo-EM, except in the case of XFELS, which is a specific type of crystallography that's kind of gaining some popularity right now. And then the supercomputing needs become just as high for much of the same reasons, because it's a pulsed source, and so you get information that's much larger in amount and has to be corrected. So it's the correcting of that information that takes a lot of time as well. Computer research.
Okay. So we'll go ahead and let's move into a little bit deeper into disease and drug design. So pharmacology, which was mentioned in the video and is the branch of medicine that studies how drugs affect biological systems and how organisms respond to drugs. So maybe Alex, could you speak a little bit to how structural biology has been useful in pharmacology?
Well, it really stitches it all together. Structural biology holds pharmacology together, all the different pieces. You begin with identifying the protein that you're going to target in order to solve a particular medical problem. That's a structural biology problem. Then you have to find out if a particular drug or a series of potential drugs are going to be effective or not. That's a structural biology problem as well. Most of the time, the answer is no. And then you're going to have to improve the best of your drug candidates in order to get an actual approved drug. That's also a structural biology problem. So from the beginning to the end, the pharmacological fundamental process of problem of finding therapeutic treatments to disease is a structural biology problem.
Yeah, and we'd seen that in the video. We saw there was a model of penicillin binding to bacteria to treat infection. So can you speak a little bit more to how techniques like X-ray crystallography help with the treatment and development of design of drugs?
Sure. So in the case of penicillin, for example, one of the problems we're having more recently is that there's emerging a whole series of penicillin-resistant organisms. And so we're trying to design new drugs that are able to overcome that resistance. For example, in the case of penicillin, you have amoxicillin and you have a whole bunch of different, they all end in cillin. They're the penicillin derivatives. So how do these drugs work, and how do we find out what the structure of these drugs will be? So I'll share my screen with you, and I think you can now see. So can you see this molecule?
Yes, we can.
Okay, so this is penicillin binding protein 5, which is a bacterial enzyme that's involved in penicillin resistance, and this is in its closed conformation. This is the same protein in the open conformation. These structures were developed at Brookhaven Lab by a group of high school and college students, Millie Ma, Daniel Adamek, and Miranda Minelli. All are local from local high schools and now going to Stony Brook and a couple of other nearby universities. So what these students did, they're young people, the age of our audience, and what they did is they grew crystals of this protein, which is involved in the development of resistance to penicillin in both its open and closed conformation, so that they could figure out how it works. And that was then used by the students by the researchers that the students were working with, in order to be able to design new drugs or new variants of penicillin that would evade this mechanism of destruction. The way that let me stop sharing here. The way that these organisms develop resistance to drugs can be many different things, but one of the mechanisms is by destroying the drug. And so what we're trying to do is design penicillin derivatives that are immune to that destruction. That's just an example of how structural biology links with drug discovery. You can't design a drug that's immune to elimination by the organism unless you know what the mechanism is that the organism uses to eliminate your drug.
Yeah, that's super important. Let's talk a little bit about disease prevention. So, John, how might structural biology help with things like vaccine development?
Yeah, so that's a really good example. And actually, I know you wanted to touch on some current events with this.
Sure.
So maybe I'll kind of tie these two things together and walk people through some thoughts about vaccine development and how we might be going about getting a vaccine for things like COVID-19. But the philosophy is the same, regardless of what you want to develop a vaccine for. So if you imagine something like a virus, there's an outside of the virus, and that virus is going to interact with some of your cells, right? It's going to try to get into those cells. And there are particular proteins on the surface of the virus that are going to mediate those interactions, okay? There can also be other proteins on the outside of the virus that might have other functional purposes. But what we really want in a vaccine is you basically want to prime your body to be able to recognize a foreign invader more effectively. Okay, and so kind of the strategy for doing that, I mean, our immune system does this naturally very well, right? The idea is with a vaccine, though, is that if you can provide the patient with some sample of what you're trying to protect against, that you'll develop an immune response against that in advance, so that when your body actually sees a real virus or a real bacterium, that it can already have a defense ready to go. Okay? So how does structural biology play into this? So if we want, I'm going to share my screen here. So in the case of COVID-19, for instance, what you're looking at on my screen here is a structure that was determined using Cryo-Electron Microscopy by a group in Texas. This is a protein called spike, which is on the outside surface of the protein. It's a trimer, meaning it's comprised of 3 units. There's one here in light gray, then a medium gray, then a dark gray. I'll spin it around a little bit so you can see it. Okay, and the reason why this can be useful in designing antibodies is that if you just have a protein sequence, you know, and you want to try to develop antibodies against that protein, there are going to be a lot of parts of that protein that are buried inside that your body, your own immune system is never going to be able to get at, to see, and to mount an effective response. So what I've done on this structure here is on one of the units of the trimer, I've actually highlighted in pink here, or in green, some bits of the protein that are actually on the surface, right? And so these are peptides, these are fragments of a protein that might serve best to be used to basically raise vaccines against and raise detection methods against. Yeah. Is that clear?
Great. Yeah.
It's
so
like you want to have the
move
right thing. You want to have the right
Absolutely.
thing that can be seen. And so that's where the structure comes in, because you're actually seeing it. And so you know what the molecule, like your other molecules, will be seeing as well.
Excellent. So the protein that you were referring to, John, is the spike protein, correct?
Correct, yeah.
So, you know, a lot of people have been obviously affected in one way or another by the SARS-CoV-2 and the coronavirus that causes COVID-19. And a lot of people now are studying this spike protein. So maybe, Alex, maybe could you speak a little bit to why it's important for scientists to know the structure specifically of the coronavirus spike protein, even aside from for vaccine purposes?
Sure. This is also a great opportunity to point out that techniques like cryo-EM and crystallography are not independent of each other. They work together. The structure that John was just showing you, I was looking at that structure about a month ago in order to decide which part of the spike protein it would be best to try to engage in crystallization trials with. So I selected a small piece of the spike protein, which the structure that John showed demonstrated is sort of optimally positioned for drug development efforts. I've got some of that protein, and now what I'm doing is crystallizing it or co-crystallizing it with lots of different chemical agents that could block it. This is very similar to what's being done with lots of COVID-19 proteins. The spike protein, the main protease, many of the different proteins, groups around the world are conducting trials or chemistry trials or chemical biology trials where they combine the protein with different potential drugs, which are often called therapeutic chemical or fragments. And then what we're looking for is to see which of these fragments will then bind to the protein, the spike protein, so that it could then prevent the spike protein from doing its job of recognizing the its human counterpart, which is how the infection begins. So the reason that it's important to be able to visualize proteins such as the spike protein is because that's the beginning of drug development, is matching or finding a way to match your protein target with a potential therapeutic chemical.
Great, and I know that both Cold Spring Harbor Laboratory and Brookhaven National Laboratory are both involved with doing a lot of research around, especially the spike protein here. John, did you have anything else that you wanted to add to research regarding the spike protein?
Yeah, I mean, the only thing that I'd like to add, and I'm sure Alex can talk more on this too, or at least verify it's true, is that, to be a structural biologist, you're not necessarily just working on crystallography or cryo-EM or NMR all the time. You also have to be somewhat of a biochemist and a molecular biologist. In our case, our lab produces a lot of different proteins so that we can do the structure determination of them. And so we've actually been recruited by a number of other labs to help generate proteins. So can we make spike protein? Can we make nucleocapsid protein to send to other labs to help them in their diagnostics development? So we don't really develop diagnostics ourselves, but you can imagine that if you're trying to do that, you need samples to test your diagnostics on, right? And of course you want to test on patient samples ideally, but acquiring those is difficult for a number of logistical reasons and safety reasons. And so when you're developing your diagnostics, you want material that you can use worry-free. And so we've been using a lot of our expertise in protein chemistry to just be a factory. Can we make things to help out other labs and help the community?
So let's switch gears a little bit and talk a little bit about kind of student careers or student interest in STEM fields or structural biology. So we have a number of students who are watching this panel discussion today who might be interested in a structural biology career path. And I know a question that I get frequently and other scientists get frequently about my career choices. Did I always want to know that I wanted to go into this field? And I know, John, you touched on this a little bit at the beginning. So either John, if you wanted to talk a little bit more, or Alex, if you wanted to address this, how did you know you wanted to be a structural biologist, and what path did you take to get here?
Yeah, I'd like to hear from Alex. I think I already kind of touched on it.
So the first time that I knew for sure, I read a book called The Still Point in a Turning World, which is a mom's story about her son, Ronan, who had a particular disorder, a medical disorder, and described the loneliness that is felt by patients and the families of patients when they realize that there are more diseases than there are people to fight those diseases. And that sometimes patients can go to the internet, just like anyone else, to see, in this case, what could maybe save her son, and find that there's very little or spotty research on particular illnesses. And I realized that the other side of that, the flip side of that, is that it means you can make a difference. It means that if you're sitting there in your chair and you're 18 years old and deciding what to do with your life, if you pick an illness and you decide to devote your life or parts of your life to addressing that illness, that you're making a real difference. You're not a drop in the ocean. You might be the only person working on that particular condition. And that means you're providing hope to people who don't otherwise have it. Structural biology is a field that has a lot of very dedicated professionals, but we are vastly outnumbered by the number of problems that we're trying to solve. I used to be a physicist, and in physics, the number of problems being solved is significantly smaller than the number of researchers. So people don't throw ideas at each other very much. In structural biology, that's just not the case. I don't mind at all. I'm very happy if someone else starts working on something that I'm doing, because it means I can devote my time to something else. There are way more problems to be solved than there are people. What that means for you as an audience is that if you devote yourself to this field, you can make a real difference. You can make a difference in the lives of patients, and you can have the feeling of knowing that your work matters, that it matters to real people. And that's how I decided that I wanted to do this.
Yeah, great. And something else that's very important, obviously, in a science field is mentorship. So we've all been mentored by senior scientists, and we've all had the chance then to also mentor younger scientists. So what have you learned through your mentoring of the next generation of scientists?
I can start with that. So I think what's really interesting about mentoring is that you often learn as much from your mentees as they learn from you, sometimes even more. I have to say, so I have a graduate student in the lab who just joined a year ago. So, maybe not quite the same age as the audience, but not too far off, right? A few years of college isn't that far different. And I'm really impressed with him and his cohort, their ability to really work across kind of different disciplines. So I think that more and more in biology, things are becoming cross-disciplinary. And that's been an evolving trend. That's been happening for quite some time. So I would say that, people in my generation have had to do more of that than people in the previous generation and before. Things used to be a lot more siloed. And I think it's really inspirational to see people being able to weave all of these different threads together to make new connections. Because like Alex said, there are way too many problems to be solved. And then you're getting a lot of creativity kind of being infused in at the same time to kind of give different perspectives on things. So that's really been quite great. And, I've received a lot of mentorship in the past about the importance of being, you can either have a broad scientific base or a very deep scientific base, and you actually kind of need to balance both of them. So you need to, pick the projects that you're interested in, the diseases that you're interested in, and study those very deeply. But at the same time, there's a huge importance in having great colleagues that are in different disciplines of biology, because a lot of times what's really interesting is you will find a problem, a technical problem, for instance, that needs to be solved, and someone has already solved it. And I work in gene regulation. I don't know too much about neuroscience, but I live with a neuroscientist, right, as a housemate. And the number of times when I have like some computational problem, he says, "Oh, in neuroscience we do this all the time. It's already solved." Right? That's huge, right? And you don't get that without having kind of all of these connections. So for the next generation, I would definitely say, like, keep your eyes and ears open and like have friends in other places.
So we have a viewer question. What kind of advice would you give to students who are interested in either pursuing a career in structural biology or maybe a research program in structural biology or other STEM field?
Do you want to take this one, Alex, or do you want me to?
Sure. There's a very simple and practical, there are a lot of more deeper answers to that, but I want to tell you one very simple practical thing about being able to accomplish that objective. There's the academic year and there's the summer. You got to use your summers. Nowadays, internships are more or less required for you to be able to make it in science. So you've got 4 years to make your academic career. You've got 4 summers in which to practice your skills, intern in laboratories, and learn and do some real science. This is just a practical thing because otherwise the door won't open. There are a lot of other things I could tell you about how to do it right, but that's the first thing I would tell you.
I would also like to add in to that, that there are a lot of great scientific careers out there. And, I love structural biology, and I've had great interactions with so many students who have come in and wanted to try structural biology, but it's also not for exactly everyone, right? And you might, for instance, if you're more interested on the pharmacology side, you might like incorporating structure, but going more into drug design, or you might like to do more biochemistry, or you might like to, get more into the particular biological question that you want. I think with all of these fields of biology that it's really important to kind of be dedicated to the questions that you're trying to solve. And it's hard to maintain that dedication if you're not really passionate about it. So I think that in addition to the internships being important for you to, kind of get the lines on the resume that you need, so to speak, to get you where you want to be, it also really helps focus you in terms of knowing what you want. If you're considering this as a long-term career path, and you might be considering graduate school, for instance, a lot of programs in biology in graduate school do what's called rotations, where in your first year or your first half year, you actually pick labs to work in for a few weeks or a few months to get a sample of which lab you like to go to. And I feel like people that do really well in graduate school already know which types of labs they want to go to before they start. As opposed to, I'm going to go to grad school, I'll use the rotation time to figure out what I want. Because in those cases, what usually happens is people pick 3 or 4 very different things, and then realize within 3 days of rotation number 1 that they don't like that lab. And now you're wasting a couple of months. You might learn some things. You might meet some great colleagues. It's not a complete waste. But in terms of figuring out where you want to go, it's not really helping you. And I think that, by doing that in high school or in early college, knowing and narrowing down which subdisciplines you want to go to, that'll really help you in the long
run. And for our viewers, there are a number of different opportunities for students to participate in a research program or an internship across Long Island. So even through Brookhaven National Laboratory, we have the Department of Energy Science Undergraduate Laboratory Internship Program. There's the High School Science Research or the High School Spark programs. I know for us at Cold Spring Harbor Laboratory, we have the Partners for the Future program, the DNA Learning Center Internship Program. We have DNA barcoding research programs. The Long Island STEM Hub, of course, has more information about opportunities for students across Long Island, and even a simple internet search. Students can find a lot of this information. So Long Island really is a hub for a lot of this technology, a lot of these STEM opportunities for students, and we certainly encourage them to go search for that information. So unfortunately, that's all of the time we have for today. So if you were viewing this panel today and you have additional questions that we were unable to address, please feel free to send them to oep@dnl.gov, and we will do our best to respond. Thank you again to John and Alex for your time. We really appreciate the work that you do and the insights that you shared with our viewers today. If you're at home and you've not had the chance to view the Branching Out with Bio episode yet, you can follow the link that we've just put up on your screen to access that video. You can also stay tuned as we'll be playing that video momentarily when this panel discussion ends. We encourage you to view the other full STEM Ahead videos available on the Long Island STEM Hub homepage. And be sure to be on the lookout for other episodes in the series that will be posted throughout the year. Finally, we'd like to thank Brookhaven National Laboratory and Cold Spring Harbor Laboratory for hosting this event today. And we encourage you to check out their programs, their research and internship opportunities, and available virtual content at the websites listed on your screen. You can also follow us on social media. So thank you, stay safe, and we will see you next time.
Yeah.
Hi, I'm Kahil. Welcome to Full STEM Ahead Long Island. You may not know it, but Long Island is a world leader in biology. Scientists and students here are making discoveries to improve how we treat diseases, make new biofuels from plants, and more. I'm at Cold Spring Harbor Laboratory. It's home to 8 Nobel Prizes, and over 1,000 people work here. Let's go see what's happening.
Cold Spring Harbor Laboratory has been here for over 125 years now. We are a vibrant research community, a lot of research labs, also a Center for Education. We have the DNA Learning Center here, a lot of conferences that come here to learn new things in biology and talk about biology.
What type of research do you do here?
I work in 2 areas. One is DNA replication, and I also work in an area called RNA interference. In general, what RNA interference is that when you express a gene is made out of DNA.
Right.
You transcribe it into something called RNA, and from RNA we translate that into proteins that are usually the workhorses in the cell and in our body. If you take a small piece of RNA that matches that intermediate RNA between the gene and the protein, you actually destroy that RNA, and then the protein cannot be made. That is used in our cells just naturally, but sometimes that gets out of control. So for example, 70% of neuroblastomas are as a result of that little small RNA not being there when it's supposed to be. And so that causes the cells to replicate in an orderly way, and so that results in a tumor. And now, using our technology of structural biology, we were able to discover which protein actually it is that destroys that RNA. So now there is a new therapy that uses this type of process to our advantage.
I'm here with Bree Bebel, a graduate student.
Well, I've always been really interested in how things work, especially things in the body. So I went into college knowing that I wanted to study biology, but I thought I wanted to be a doctor. And we're using this textbook, and there was all these references about Cold Spring Harbor Laboratory, and it just sounded like this amazing place where so much cool science had happened. When I came here, I knew right away that this was a place for me.
What does the future hold for you?
I'd really love to be a teacher, especially for undergraduate students, and really get to share with them how to work in a lab and have them help me with my research experiments.
Before I came here, I was in Germany where I did my PhD. So I was kind of following Joshua Torr's lab and the kind of work she does here. But I feel like it's an amazing place to be. One, of course, it's in terms of science. Also, the nature, it's amazing here in Long Island.
So what's the next step for you?
Back in India, there are like more people who kind of needs this kind of information because it's still developing. If I can go back, I can probably contribute much more in Indian society, Indian scientific society.
Are there special kinds of tools that you use to do this work?
We use something called Cryo-Electron Microscopy. We have a really high-end machine here at Cold Spring Harbor Laboratory, and that really enables us to look at particles of molecules and really study them in atomic detail.
Is this the only tool to use to do this kind of research?
We've actually been using X-ray crystallography for a long time, and we are really very, very fortunate to be close to Brookhaven National Laboratory and the National Synchrotron light source, the second one now that I've been using. It's really an amazing resource where we can get high-resolution pictures of the molecules that we study. That's really, in some cases, the method of choice for us.
Limor, thank you for your time today. Now let's head east on Long Island's Innovation Corridor to Brookhaven National Laboratory, where Aleida will give us a close-up view of the National Synchrotron Light Source 2.
We are at Brookhaven Lab at the National Synchrotron Light Source 2. This machine is so big that Yankee Stadium can fit in it. Let's go inside and meet the people who use it.
So this machine uses a method which we call X-ray crystallography. This uses X-rays to determine this three-dimensional structure. And we get images not like a light microscope, but we get the images by having the X-rays scatter off the crystal of these molecules. These pictures are recorded on these kinds of detectors. They are like a big camera, and then they are transferred to a computer where we do the reconstruction of the electrons in these molecules which originally scattered the X-rays. That is the method by which you then obtain the three-dimensional structure.
So why is that important?
In a way, you can understand how these proteins function a little bit like playing with Lego. So they work by molecules fitting on top of each other or inside of each other or not fitting. A good example are antibodies, which do or do not fit onto the outside hull of a virus to then help your immune system to attack them. Another good example are drugs. So when you want to inhibit a molecule and thereby kill a bacterium, you can do this by having a drug that blocks a very important function of this protein. Then this virus cannot replicate, and that way it's basically dead, which makes you healthy. Let me show you at the beamline. We have two students from Long Island high schools who are currently looking at the images that they took in their last shift.
So can you tell me a little bit about yourself?
Well, over the summer, I actually did a workshop at Brookhaven Lab. It was the structural biology workshop. I realized I have a love for structural biology, and I tried to find further programs I could further the research in.
What kind of research you've been doing here?
We're researching anaplasmosis right now, and it's carried in ticks to cause diseases in humans. And by using X-ray diffraction, we can understand the structure and hopefully help cure these diseases. After grad school, I want to become a pharmacologist. Pharmacology is like a research into how drugs work and how they affect the body. Brookhaven Lab actually is what fueled my love for pharmacology, because before I even knew anything about Brookhaven Lab, I didn't know anything about pharmacology.
I'm currently researching the protein bovine thyroglobulin. It actually is very similar to this oligosaccharide, a compound called alpha-gal, and when transmitted by ticks, this compound can cause the red meat allergy in humans, which is very detrimental. At this time, bovine thyroglobulin has no known structure. We're hoping to find a structure so we can find out more information about how it interacts with other proteins and its function.
What have you learned by doing this?
First of all, we've learned basic lab skills like micropipetting, and to be more specific about protein crystallography, we've learned how to create crystals and fish crystals and even run the beamline. Before I started this research program, I was dead set on becoming a doctor, but now I'm gravitating more towards the research. It's such an honor. I never thought I would be in this place that I am right now. I love working with the scientists, and I've learned so much from them.
There are so many career opportunities in the life sciences here on Long Island, supported by world-class institutions like Cold Spring Harbor Laboratory and Brookhaven National Lab. To learn more, visit listemhub.org.